Good evening, everyone, and welcome to the webinar. My name is Tanya Winders, and I'm the President and Chief Executive Officer of Allergy and Asthma Network, the nation's leading nonprofit dedicated to ending needless death and suffering due to asthma, allergies, and related conditions. Tonight, it's my pleasure and honor to welcome you to the webinar, The Why Behind Latex Allergy and Cross-Reactive Foods, which is presented in coordination with the American Latex Allergy Association in honor of Latex Allergy Awareness Month. Furthermore, I'd like to say a big thank you to our presenting sponsor of tonight's webinar. An educational grant was provided by Mylan Specialty, the manufacturer of EpiPen. And I'd also like to thank our co-host, the American College of Allergy, Asthma, and Immunology. This evening, we are honored to have with us our presenter, Dr. Tom Greer. Dr. Greer received a BA in biology from State University of New York at Plattsburgh and his Master's of Science and PhD degrees in chemistry from The Ohio State University in Columbus. From 81 to 83, Dr. Greer performed postdoctoral research in the Department of Chemistry at Cornell University. Between 84 and 93, he was a research investigator in the Allergy and Infectious Disease Immunology departments at Abbott Laboratories, where he led teams involved in analysis of antigens and antibodies. Dr. Greer joined Greer Laboratories in 1994 and currently oversees research in several areas, including development of extraction products and allergen-specific quantitation methods and stability, compatibility, and cross-reactivity, the topic of tonight's webinar. He continues to publish scientific papers in a wide range of biochemical and medical journals and present the results of his group's original research at regional and national allergy meetings. As we get started this evening, if you would like to pose a question throughout the webinar, you may go to the control panel and raise your hand or ask the question, and we will field that for you all. The lines will be on mute with the exception of our presenter and myself as the organizer until the end of the call when we'll open the line for additional questions. At this point, I'd like for all of you to join me in welcoming Dr. Tom Greer.
Thank you, Tanya. I appreciate it very much. And I'd like to start out by thanking Tanya, Brenda, and Sally at the Allergy and Asthma Network for helping to coordinate this webinar, for Sue and Marcia at the American Latex Allergy Association, for all that they do to support efforts to inform all of you on new developments in latex allergy, and of course to Mylan and the ACAAI for their support. This is an intriguing subject for me. I've been interested in it for many years, and it's been a subject of considerable interest to the allergy community for the past 25 years. And because of research in this area, which continues today, we now know that about 2/3 of natural rubber latex sensitive individuals carry specific IgE antibodies to some type of fruits or vegetables, and that 1/3 of these are actually symptomatic after eating these particular foods. Most of the symptoms occur in the oral cavity and are reasonably mild, but in some cases they can be severe and can cause anaphylaxis. And for the next 45 minutes, I'll be commenting on a variety of different subjects related to latex and cross-reactive foods. And I hope my comments will help you understand why these reactions occur, what causes them, and what you can do to help recognize if these relationships are important to you or to others close to you. And I will do my best to answer 5 fundamental questions tonight, which are listed on the next slide. Some of these were covered last night in Dr. Sandra Gottschalk's presentation, which was wonderful, and I hope you got a chance to listen to Sandra talk about latex allergy. I'll talk briefly about what is latex, where is it found, what types of plants contain latex, what's its function in nature, how do latex preparations vary in terms of their chemical and biochemical composition, and then the big question, of course, is why do so many common fruits and vegetables show these allergenic cross-reactivities with natural rubber latex products from latex derived from both Asian and Central or South American rubber trees, the Hevea brasiliensis. And there will certainly be time at the end of the webinar for your questions as well. We can go to the next slide. So what is latex exactly? Well, I think we all have seen pictures of the trees that have been cut and the latex that's oozing from the trees into the buckets the way it was collected. Dr. Gottschalk had a slide on this last night. It's considered the sap, but there's actually another fraction in latex that's considered the sap or the resin that has a slightly different function. This is the white, milky white fluid that is present in stems, leaves, and in the bodies of a variety of different vascular plants. It's actually present in elongated cells or canals called laticifers. It's a hard word to say, but it's the actual term. And in most plants, these latex-containing channels are analogous to the blood and lymph systems that we're familiar with in humans and mammals. It's typically white and milky, opaque in appearance, but in some plants can appear to be clear, sort of a light color with an orange tint, and somewhat translucent. The chemical compositions of the latex are very complex, and we'll talk about that as it relates to both its natural function and allergic reactions. On the next slide, we have a listing of what types of plants contain latex, and literally thousands of plants, both vascular, which have flowers on them, and non-flowering, mostly conifers, species from a wide variety of unrelated plant families. The current count is somewhere over 20,000. So you can bet that virtually every plant or tree that you come in contact with may have some form of latex in it. And I'll talk in a minute about why that's important to the plant. The latex-containing plants tend to be much more common in tropical areas, typically Central and South America and Southeast Asia, compared to the more temperate North American regions where many of us live. Most of the products, most of the latex that I'm familiar with comes from Guatemala and Central America, and in Malaysia and Southeast Asia. The best known sources of latex are listed at the bottom of this slide. Most of these are somewhat familiar to many of you, I'm sure. Poppy and milkweed are very common. We've probably all seen them out in the fields. The weeping fig tree is another source of latex that does seem to have some relationship to the natural rubber latex. And then the last one listed is guayuli, or parthenium, which is an interesting discovery that latex from that plant has no relationship to natural rubber latex. So guayuli is now beginning to find itself into a lot of products as a substitute for natural rubber latex, and it's found to be very safe for individuals who are sensitive to the natural rubber latex allergies or allergens. What is the function of latex in plants? It's very simple but very complex in terms of how it works. It's the primary defense mechanism that plants have to counter attacks mostly by insects and microorganisms. And these defense mechanisms act in a number of different ways, some physical, some chemical, and some based on the actions, the catalytic actions of enzymes. They appear to have evolved independently, but in very similar ways, what's called convergent evolution. So these plants, many of which are completely unrelated to one another, happen to develop the same types of defense mechanisms completely independently and converged in very similar ways. And there's a very interesting give and take historically on how both the plants and the animals have adapted to changes in the content of the defense mechanisms in order to counteract it and find resistance. And so there's I don't have time to go into that today, but it's a very interesting example of how organisms change to survive, and it's defined in latex on a very specific molecular and metabolic level. It's evident from these compositions and properties of latex that these relationships are very important to the plant, and they have a direct relationship to allergy. Another aspect of this that you may not have thought about is that these defense mechanisms are elevated when the plants are under stress, and that can be from a severe growth condition or climate, exposure to chemicals. And what will happen is these particular defense chemicals will be stimulated to be more highly expressed under conditions of stress. If these particular compounds happen to be allergens, then these plants can also be considerably more allergenic if they've undergone a certain degree of stress. And if you think about what genetically modified organisms are, these are plants that have been bred to be much more resistant to stress. And so they have this obvious high agricultural value, but they could also be quite a bit more allergenic. And the allergy community is paying very close attention to the types of changes that are occurring in genetically modified organisms, in particular those that involve defense mechanisms and defense-related chemicals, such as those that we'll continue to talk about this evening. Next slide. How are latex fractions isolated? Sandra talked about this briefly last night, so I won't go into much detail. Again, the rubber trees are cut or scraped, the milky fluid is collected, and then it's centrifuged at a very high speed. And what you get when you do that are three fractions. You get a top layer that's buoyant, where the rubber particles float. You get a bottom layer, which is sediment, into a pellet, which is called the B serum. And what's left is this clear, fairly viscous liquid in the middle, which is called the C fraction, or the C serum. Most natural rubber latex products are made from some combination of the rubber particles and the C serum. And the sap gets its milky appearance because the rubber particles suspended in the clear fluid give it that milky suspension, very much like bovine milk is a suspension of particles in a clear liquid. Next slide, please. So how similar are latex preparations from different sources? This is a complex question, and one that really gets down to the crux of what latex does. And it really is dependent on similarities between chemicals in three different categories in these latex preparations. The particles that we just talked about that are isolated when you float they float on top of the preparation after centrifugation. In the C serum, there are toxins and metabolites that are critically important in the defense mechanism. And then there's a series of proteins and enzymes where most of the allergens fall. Now, the particles are involved directly in defense mechanism. When an insect, for example, attacks a leaf on a plant and breaks the surface, the latex will ooze out of that hole that's created by the insect, and the particles will initiate a gelling process. So basically what happens is the insects get trapped and they get stuck in that particular spot where they've damaged the leaf, and it immobilizes them and eventually kills them. Another mechanism that's independent of that are that these toxins and enzymes in the different latex preparations are fairly specific for the exact predators that act on those particular plants. And to some degree, the plants have evolved and the toxins have evolved to match the predator, again, to increase its potential for survival. These are mostly insects and molds. And the enzymes that are present in the latex liquid also selectively attack the outer surface of these predators. And you'll see attacks against chitin, which is a major structural protein network on molds and to some degree on insects as well, and complex sugars or glycans, which are very common on the outer surface of many mold organisms. And so you'll see as we move forward, there will be terms like chitinase and glucanase. These are the specific proteins that are in latex that counteract these particular structures on molds and insects and help the plant survive from attacks from these organisms. The relevant characteristics in the latex preparations are the allergens. They're found primarily in the C serum, but there's some that are found on the rubber particles. We'll talk more about this shortly. And again, these are the fractions that are used to manufacture medical and consumer products. Their distribution, function, and activities can be shared with those in either botanically related plants or those with similar natural predators. So if there's two plants that happen to, in genetic terms, be completely unrelated, but the same type of predator attacks them, they may have evolved these independent mechanisms to end up with very similar compositions in their latex preparation to help the plant defend itself from these attacks. There are relatively low levels of these releasable latex allergens in molded products versus the dipped products. Sandra talked about the dipped products last night. The easiest way to remember this is that dipped products tend to be soft. The balloon is relatively soft. It's pliable. And in those types of examples, the latex allergen is usually very easily released. And we know from gloves, for example, that both sweat and the cornstarch powder can remove latex proteins and latex allergens from the glove. And as soon as the glove is snapped and the powder is airborne, it's aerosolized, and that becomes a major sensitizer for people who are sensitive to that. A molded product would be more like a vial stopper for a pharmaceutical drug. And the analogy that I like to use for the molded, where the allergens aren't really released very easily, is think about a marble that's embedded in concrete. You can see that it's there. It's obvious that it's present, but it's stuck and it's not going anywhere. It's certainly not coming out without a major effort to release it from its contact in that cement or in that concrete. That's what vial stoppers are. There are some reports that latex allergens can be released from vial stoppers, even if the stoppers are made with latex. And while the potential exists for that, the levels are much, much lower than those that you might find in products that are made by this dipping process. On the next slide, I want to take a minute and talk about allergen compositions. This is relative to latex and any other allergens that you or your loved ones may be sensitive to. A typical allergenic source contains over 1,000 water-soluble components. Many of those are large molecules like proteins and complex sugars, but there's a variety of small molecules as well, metabolites, pigments, and salts that make up most of the the metabolic components in these different sources. Of these 1,000 components, about 50 to 100 are capable of producing an immunological response or an immune reaction based on their structure. And typically 5 to 10 proteins in each source or extract, the products that my company makes, are usually responsible for allergic symptoms in sensitive patients. Now, if all of us reacted to the same set of 5 to 10 proteins from a given source, such as latex, the diagnosis and treatment of these diseases would be much simpler, easier, and frankly better. Unfortunately, for most airborne allergens and environmental allergens, this is not the case. On the next slide, I'll show you the way that specificities are usually represented. These are charts showing dust mites in grasses where there are between 18 allergens for dust mite and 13 allergens for grasses that have been identified. Based on a number of clinical studies and laboratory studies, we can now estimate the percentage of allergic patients that show sensitivities to each one of these particular allergens. For dust mite, for example, you can see that mite allergen number 1, and these group allergens are interesting because across different dust mites or across different grasses, these allergens share a lot of very similar properties. And we'll talk in a few minutes about homology and identity of sequence, and this is where that comes into play. So if you look at an allergen like dust mite allergen number 1 on the left side graph, you'll see that there's about 75% of patients recognize that allergen, and it's considered a very important allergen clinically. But 75% means that 1 out of every 4 people don't recognize that particular protein. They may recognize allergen 2, they may recognize 9, or they may recognize a subset of those. The same for the grasses. Allergen 1 and allergen 5 are the most common, but there are some folks that don't recognize those particular allergens and are sensitive to a different group or a different collection of specific allergens. So understanding these profiles can definitely help us improve the accuracy of testing and the effectiveness of treatment, and in many cases, clinical decisions on the best way to manage or minimize allergic reactions. The potential, we can go to the next slide, the potential or the hope in looking at profiles is that specific groups of allergens can be recognized by and defined for subpopulations of sensitive patients. And the good news with latex is this is indeed the case. And different combinations of the 14 allergens that are listed on this slide define the different diseases that are related to latex sensitivity in the different populations. And we'll talk more about this toward the end of the webinar. But it's safe to say that the discovery of these allergen profiles for latex has dramatically changed the way that we view and manage latex allergies. And a lot of these are now inherent in the way latex allergy is diagnosed. And I think you'll see as we move forward where this becomes important in clinical decisions. I want to take a minute and talk about cross-reactivity because that's the essence of the seminar, or the webinar tonight. Cross-reactivity basically means that there are very similar, what's called homologous or identical is maybe a simpler word, protein allergen sequences and three-dimensional structures that are recognized in a very similar manner by the allergy-dependent IgE antibodies in sensitive patients. These are typically, these protein allergens with similar structures typically have very similar functions. There's a very close relationship, as you can imagine, between structure and function and activity for many proteins, and these fall into that category. So highly conserved functions, molecular activities, and in many cases allergenic properties. And for latex, we now know that the affected populations or groups, different groups, possess very different profiles, specificity profiles, and coincident to that, different sensitivities to various foods and pollens. On the next slide, I have a very crude diagram. This is one of my favorite diagrams to talk about in seminars. This is an old diagram of the three-dimensional structure of a protein. There are two proteins shown here. They're both proteolytic enzymes that are very common in nature. Elastase on the left and chymotrypsin on the right. They have similar functions, but their structures are quite different. I've highlighted in the green areas those sequences that are absolutely identical between the two proteins. And it only adds up to about 40% of the molecule. But if you look closely, the middle of the molecule has what's called the active center or the active site. That's where the enzyme really does its job. And in forming that active center, most of the regions that are similar or homologous, shown in green here, are used to actually form that binding region. As you move toward the outside of the molecules, you'll see more and more white, and you'll see different types of loop structures. In most proteins, the allergenic sequences are toward the outer end of the surface. So these two proteins would probably not cross-react very highly because they only share 40% sequence identity, and most of it is on the inside of the molecule, not the outside. So as the relationship between these proteins gets closer and closer, the structural similarities become more and more precise and more similar, and the degree of cross-reactivity increases. For an allergen to be cross-reactive with another protein, you typically have to have about 80 to 90% sequence homology, which means that a lot of these outer surface sections have to be virtually identical or similar enough to where the antibodies recognize it as similar. So on the next slide, so what we're talking about here, in order for two proteins or two entities to cross-react, there has to be multi-point complementary interactions in 3 dimensions between specific groups on the antibodies and the allergenic molecules. And it's very much a lock and key situation. We all have had keys that fit into locks but don't turn the tumblers. They're just a little bit different, but that difference is important. And that's a good analogy for this interaction. So you can have optimal combinations and you can have minimal combinations, but typically there are somewhere between 15 and 20 amino acids on each molecule that are involved, some continuous and some fairly distant. So it's a very interesting situation to when you're studying cross-reactivities to look at the structures and try to deduce exactly which regions are contributing to those particular reactions. There's only one way to confirm cross-reactivities, and that's in the laboratory using a procedure that we use in my lab. It's very similar to this format, and I won't go through this in detail, but the simple approach to this is that you take a solid phase, which in this plate is a plate surface on the left-hand side, and you immobilize an allergen mixture on that. And so in this case, it would be latex. And then you add a serum antibody with a second extract or a second component that you're looking to see if it's structurally related to the one that you have on the solid phase. In this case, for example, let's say we add banana to the solution with the antibody in a patient that's cross-reactive with latex. The banana and the latex components will compete for that antibody, and the more of the cross-reactive component we see, the more of it binds to that antibody, and the less of it ends up binding to the next probe, which generates color. So what we can do in an assay like this is quantify the amount of color that we see, which is directly related to how much inhibition we see. And if we go to the next slide, we can determine how similar or different the two test materials are based on both the magnitude of the inhibition and how parallel they are. So there's both a qualitative and a quantitative aspect to this. Sometimes you can have a few components in the mixture be similar and then the rest will be different. And those will tend to look like the purple line at the bottom, where you do see some reaction taking place, but it seems to be different in terms of its dose response characteristics compared to the reference material, which in this case would be latex. There are some very general patterns that have been established for cross-reactivities in many plants, and these apply to some animals as well. Two items within the same genus or tribe are typically strongly cross-reactive. They share closely related protein compositions and structures. Two items within the same subfamily are typically moderate. There are some similarities and there are some differences, and the degree of cross-reactivity can vary quite a bit from one patient to the next, depending on which particular molecules they recognize. And then two species in different subfamilies typically have low or no cross-reactivities. They're almost completely different immunologically, and so those tend to not be a significant contribution to both the laboratory or clinical reactions. On the next slide, I've listed some of the major food allergens, and I added heat stable to the title because what we've found with food allergy is that most of the allergens that are the prominent allergens in both kids and adults are very stable to heat, which means they survive cooking, they survive roasting, heating, and they survive the stomach acids and the enzymes in the stomach that would normally inactivate many other allergens. The list I have on the left side of this slide accounts for about 95% of food allergens, according to recent surveys. If you look at that list, you'll recognize that none of the foods that we normally see associated with latex are on that list. So most of the foods that seem to have a link to latex are relatively minor, and it turns out that they're unstable to heat and they're unstable to digestive enzyme, and that becomes important as we try to both diagnose and manage latex food cross reactions. We can go to the next slide. There are a number of these food cross-reactivity syndromes that involve proteins from both related and unrelated plants that share common functions. In most cases, the primary sensitizers, the entities that the patients are truly allergic to, is either the pollen or, in this case, natural rubber latex. And then they react, the individuals will react to foods that happen to share common structural cross-reactive proteins with the primary pollen or the latex. Again, these are relatively minor food allergens. And not saying that, you know, if you have these sensitivities that they're insignificant, but in the big picture of food allergy, they're actually considered to be like incomplete allergens because they don't they can elicit symptoms, but they typically don't sensitize. And they're found in a number of categories, and I'll talk a little bit about the first two, plant defense or pathogenesis-related proteins, which, as you can imagine, is common in latex, and profilins, which are common structural proteins found in many plants. So the pathogenesis-related proteins, on the next slide, there's 14 families of this. It's the same number as the number of latex allergens, but that's just kind of an interesting coincidence. And as you look at these families, you'll start to see some of these names that we talked about earlier. Beta-glucanases for PR2 that contains, of which Hep B2 is one of the members. The chitinases for groups PR3 and 4 that have very similar structures to the Hep B6, 0.02 and 0.03 latex proteins. There's a chitinase that's in PR8, and there's a lipid transfer protein, which is another plant defense series of proteins that's important for latex. Before we get into the latex-specific ones, I want to take a minute to talk about the PR10 that's shown in blue. This is the birch pollen homologs, or the birch the pollen food cross-reactivity syndrome that's very common in a disease called oral allergy syndrome. We can go to the next slide. This is a fairly old association that was first noticed between birch pollen and apples around 60 years ago. The situation was folks that had pretty severe birch sensitivity just could not eat apples and couldn't tolerate apples, and it took some time to make the association that they actually shared some proteins. They share these homologous allergens. They're related genetically in some cases, but not all. But in all cases, the pollen sensitivity precedes the reaction to food. It's not a food allergy that's exacerbated by pollen. It's actually the opposite. The typical foods involved in oral allergy syndrome are birch, mugwort, which is a weed, and hazelnut pollen. And the typical foods are apple, celery, and carrot. And there are specific proteins that are part of these profiles. And there are actually different protein groups for the birch celery versus mugwort celery cross-reactive syndrome. So we know a lot more about these particular, these particular syndromes, but they're very different from the latex food syndrome in two important aspects. One is that the latex cross-reactivities are much more prevalent, but more importantly, latex cross-reactions are much more severe and can have much greater risk at having the patients go into anaphylactic shock because of the association to latex. But I wanted to mention this, and the next slide just has a little bit more information. I think I'll skip through this one very quickly. The one thing I would like to say here under the third point is isoallergens. There are a lot of different varieties of food, and the structures of the proteins, the same protein, from a different variety of food can sometimes be very different. And in some cases, those differences can change the allergenic character of this cross-reaction. For example, there are some apple varieties where the corresponding protein in apple, which is called MALD1, has several differences from the major birch protein, and those differences are enough to destroy the cross-reactivity. But if you look at another variety of apple, the structure will be more similar and the cross-reactivity will persist. And so it's difficult sometimes to predict or to just label something as an apple allergy or a birch allergy when the actual specifics of the disease are much more precise than that. And we're beginning to be able to define these relationships in these much more specific terms. And your physicians have more tools available to them today than they did 5 or 10 years ago, and hopefully in 5 or 10 years we'll have even more, to where they can define your allergy based on not what you're sensitive to, but the specific proteins that you're recognizing. On the next slide, we have some information on the lipid transfer protein family. This is relevant to latex. Again, it's a plant defense protein. It has different structural characteristics and some different pollens and foods than we've seen with the others. The interesting thing about lipid transfer protein is that these are patients that show no birch or no grass pollen sensitivity. And they're characterized in many cases by a strong reaction to plums. They have difficulty, severe difficulty digesting or eating plums. Plums are very high in this lipid transfer protein, and they're very low in the allergens that would correspond to the other birch families. And the risk for anaphylaxis with these lipid transfer protein reactions is much higher with sensitivities to LTP versus the PR10s that we talked about before. Most of the LTPs are on the outer surface of the fruit, so if you're only consuming the pulp on the inside, you're probably okay, but most of us would probably eat a plum whole and be exposed to these, and they wouldn't necessarily come off easily during washing, so they might persist on the fruit itself when we ate it. And some severe reactions have been associated with sensitivities to just the lipid transfer proteins. On the next slide is the propylene, which is another family that's important for latex. These are cytoskeletal proteins. They're present in virtually every plant that's ever been studied. There's a latex protein, HepB8, which is a propylene and is important. This shows up a lot in blood tests when you're looking at the IgE reactivity of patients. It's a relatively minor allergen, only present in about 20% of latex-sensitive individuals, but it can be an important food allergen. So on the next slide, the next few slides, to kind of finish up the talk, what I want to do is actually put the profiles together for you. And there are three major groups of latex-sensitive individuals: healthcare workers, which are shown on this chart as HCW in the fourth column from the left; those with multiple surgeries or spina bifida patients in the column just to the right of that; and then symptomatic natural rubber latex sensitive individuals, which may be some or many of you. The healthcare worker population reacts to the allergens that are highlighted in green: HepB5, HepB6, 0.01, 02, 03, and HepB13. And if you go across to the right-hand column, you'll see the specific foods that are associated with, or very commonly associated in terms of containing cross-reactive components to those particular allergens. And you'll see the common high-risk players in here: banana, avocado, chestnut, kiwi, and a variety of others. So if you were a healthcare worker and you were having reactions to foods, these are the specific proteins that in all likelihood were involved in your reaction. If we go to the next slide, the next population is the multiple surgery or spina bifida group. And these individuals are also sensitive to HepB5, like the healthcare worker group. They are not as sensitive to HepB6 or HepB13, but they're extremely sensitive to the proteins HepB1 and HepB3. These are the two proteins that are predominantly present on the rubber particle itself. And so this particular specificity profile would be important for individuals who would come in contact with latex products that are made with a combination of rubber particles and C-sera. And there are a number of products that are likely to be made in that manner. The HepB1 and HepB3 are present in C-serum, but at very low concentrations. And so if it's a C-serum-based product without the rubber particles there, chances are fairly low that these particular allergens would come into play. There's not many associations with food on the two rubber particle proteins. The only one that I could find was kidney bean that's been associated with HepB3. On the next slide is probably the most important group for all of you, and that's the symptomatic natural rubber latex sensitive population. And you'll see that we have, again, HepB5, HepB6 with the major players involved, but we also have two others that are unique to the symptomatic population: HepB2, which is one of these glucanases that we talked about earlier, and the lipid transfer protein, the PR14 group, which is HepB12. And again, you'll see some of the familiar foods, cross-reactive foods, on the far right-hand column. And so we now know for, in many cases, that these are fairly unique to the symptomatic latex population. And we're learning more and more about the specific concentrations in these particular foods and variations in terms of different cultivars or varieties. But again, remember that these, in many cases, are stress proteins. And if these plants are being bred to be more resistant to stress, there's a chance that more of these molecules could be made, and they could be important clinically in terms of provoking reactions. The last group really doesn't have much association with latex. And this is really the true food allergy group. And these are proteins that happen to have some relationship to these true foods. Now, interestingly, if you look at the profilin group that have B8, you'll see a lot of the same plants, fruits and vegetables, that you see up above. So this is part of the reason why it's so difficult to make a definitive diagnosis on latex allergy. You have a lot of players. You have this duplication of both molecules and sources to where it's not a neat, clean, and easy diagnosis to make. We're getting to the point where the laboratory tests are getting better and better. But as we discussed last night, as Sandra discussed, we still do not have a latex skin test allergen in the U.S. My company worked on that several years ago, and there were some, I think, pretty interesting reasons why that was difficult then and why it would be difficult now. And honestly, I'm not even sure it would be that helpful because depending on which population you fall into, you may actually need a different type of extract to address the specific allergens that you're sensitive to. And so, as we continue to move forward to try to find more appropriate diagnostic approaches, I think we were definitely considering what we can do on the skin test side, but I think knowing what we know now, we really have to make a much more concerted and specific effort to develop products that specifically address these known populations. And I think if we do that, then we'll serve the medical community much better and make a much more accurate tool for doctors and nurses to use to diagnose latex allergy and food cross-reactivity. On the next slide is the first part of a bulletin that I wrote for the ALAA back in 2009. If we can go to the next slide, Tanya. Thanks. At that point, we had about 44 foods identified on this list of cross-reactive foods with latex based on either clinical or laboratory studies, and these were also based on publication, not just citations on the internet, but true publications and the peer-reviewed publications in the scientific literature that showed that there was some relationship, some clear relationship between these products using methods similar to the one I described earlier, the laboratory inhibition method. We can now update this. Since 2009, there are, I have on the next slide, 11 additions that we could put into the low or undetermined group. And Sandra came up with a 12th last night that I confirmed this morning, and that's the goji berry or wolfberry that we can add to this list under the fruits. That's a berry that is actually in the same family as some of the other cross-reactive foods. It's in the same family as potato, tomato, peppers, and eggplants, so it's not a surprise to me that it would also fall into this category. That is everything that I wanted to cover tonight. I'll be glad to answer any questions that you have. I know we have some time here. I do want to say before we get into the Q&A, that one of the questions that I can anticipate is, what can we do, or what can you do to help identify if a particular fruit or vegetable is relevant to you in terms of cross-reaction? And there's a couple things that can be done. There are blood tests, and Sandra mentioned some of these last night, that are specific for these particular latex allergen components. And the for healthcare workers and symptomatic natural rubber latex sensitive individuals, there are tests available, blood IgE tests for Hep B5 and the 2 Hep B6 isomers. Those would help identify, and you would know, obviously, if you're a healthcare worker or not, whether that's a relevant population for you. For spina bifida patients or multi-surgery patients, Hep B1, 3, and 5 are available for testing. And for the true food allergic patients, Hep B 8, 9, and 11 are also available. And as Sandra said, these tests are good. They're not perfect, but they're good, and they're getting better. And it provides, again, if you're mildly sensitive, the tests may be negative, but if you have a moderate sensitivity, these tests are really good at picking up your sensitivity based on IgE testing. The other option that you would have, or your physicians would have, is there's a technique that some allergy clinics use called prick-to-prick skin testing. And what they do is they'll actually take a skin test device and they will prick the actual food that you may be sensitive to. For example, let's say pumpkin in this case. They will apply a little bit of the pumpkin meat itself onto the skin test device, and then transfer that to your skin and gently prick your skin with the whole pumpkin. It's not an extract, it's the actual food. And in these cases, because a lot of these fruits and vegetables are relatively soft or can be made soft, as long as you don't heat them, which, as we talked about, could affect the allergenicity, you can get a fairly reliable skin test result from that. And in a lot of cases, foods that don't exist currently as licensed extract products can be tested in this way. And there's more and more clinics doing this. If you go to an allergist now, definitely ask your allergy clinic if they're familiar with prick-to-prick testing, or if they do it in their clinic. It's still not a highly common procedure, but it's becoming more common as its value is being better publicized. And so I would encourage you to find out more about that. I wouldn't, definitely not. I'm not a medical professional. I'm not a doctor of that type. So I can't really recommend that you do this type of testing on yourself. I would definitely not recommend that because of the obvious risks. But in the hands of an allergy or an ENT clinic that's familiar with this technique, they can, in many cases, very simply and easily and in a fairly painless, while you're in the clinic, help you identify if you're sensitive to these particular foods. Again, it's just another option that you have to help answer some questions.
Great, wonderful. Thank you so much, Dr. Greer. That was so informative and again, so interesting to learn more about how latex does cross-react with so many different foods. We do have several questions in the queue, so let's move on to those. The first is, my husband, who does have a latex allergy, recently developed an allergy to unpeeled peaches. He is a surgeon. He does not use latex gloves because of his serious allergy. He does use polyisopropyl. So should he be tested for latex allergy?
If it's, I guess it depends on the type of reaction that he has, because as Sandra mentioned last night, there is also a contact allergy that can be associated with latex, and to some degree, some of these outer surface proteins could be contact allergens. But my guess is if it's a peach and it comes from the peel, then it may be one of these lipid transfer proteins. And if that's the case, then it may be that only those particular foods that are high in lipid transfer proteins or have these LTPs that happen to come off the surface fairly easily might pose a problem for him. It's possible that if that's the only protein that he's sensitive to, then chances are he would probably have a mild latex sensitivity, but I wouldn't think it would be severe. I would think the food allergy reaction might be more of an issue than the latex allergy. But if he had, it would be interesting to see if he did pursue this with a blood test, since Hep B8 is one of the antigens that's available on the blood test. It would be interesting to see if any of the other latex allergens would also come up positive. That would be suggestive of maybe a more significant clinical allergy to latex compared to the food allergy to the LTP.
Okay, wonderful. Thank you. Next question, is the NRL allergens and specific profile chart available to share?
Yes, those are actually very common in the biomedical literature, and anything that I've provided on the webinar tonight can be shared.
And of course, we will make the playback of this evening's webinar available to you all in a follow-up email after this evening. Next question. Thank you, Dr. Greer. Excellent presentation. Right now, I am down to 8 to 10 foods that I can eat. How can we get copies of these specific profiles, which we just shared, that we'll be sending them out? Any other comments about that limited of a diet at this point, Dr. Greer?
The only comment I have is I hope you have a dietitian that you're working with closely. Most dietitians are now very aware of food allergies, and hopefully they're aware of the relationship between food allergies and latex. Have a frank conversation with your dietitian about food allergy, and my guess is that they're probably very informed. I know some dietitians in this area are very up to date on their knowledge of food allergy, because it has changed fairly extensively in the past 10 years or so, and they have to keep up, because, and I think part of that too is the changes that I talked about earlier with genetically modified foods. If you're a dietitian now, I don't think you can ignore the potential consequences of either the GMOs or the possibility that these modified foods could be a health risk to some of their patients. So usually that community, and I know perhaps Tanya and Sue can vouch for this too, that there's a very high awareness in the dietitian community on food allergies, and I think they're doing great things to help manage difficult situations. And I'm sorry to hear that you're so restricted, but I hope you're working with a registered dietitian, and if you are, then I think you're in great hands.
Great point. Thank you for making that. Next question. Why is there no skin test currently in the U.S.? This individual says that they've been told by really good sources that FDA was quite obstructive even when we were working on this and trying to advance the cause.
Yeah, I work for a company that deals with the FDA every day, so I don't want to say a whole lot of bad things about the FDA. I will say this. I think the FDA in our field does its best to be very fair and balanced, and try to be an appropriate arbiter of what's licensable and what's not in this country, based on both safety and efficacy. Their primary role is to make sure that any product that hits the market is very safe. And we know from a lot of news stories lately that, a few years after certain drugs enter the market, new developments appear to where side effects are now much more severe than they were believed to have been during the pivotal trials that were used to market the product. I think it's a very thankless task that the FDA has. It's a very challenging task to meet these standards, but I do have to say, because in the U.S. everything that gets marketed and gets distributed has to go through this process, that the safety level of the drugs that reach you are very, very high. In Europe, they're usually allowed to hit the market before a lot of safety data is determined, and there's a window of time to where the clinicians in Europe can use these products and use them, to their best abilities, so they can get on the market, but they don't carry this safety profile, and in some cases there can be, some fairly catastrophic outcomes from the use of these products because they haven't been vetted in this way. So that's sort of the politically correct answer, but I will say one thing about the specific experience that I had working with the FDA on the Greer latex skin test allergens. There were really two major challenges that we just could not seem to meet. One of them is apparent from the last few slides of my presentation. Different populations of latex-sensitive individuals recognize very different groups of proteins. And for one extract to work for all of them, we would have had to have had an extract that had appropriate concentrations of all of those different allergens in order for a single latex allergen, skin test antigen, to be appropriate for all those groups. And that just didn't happen. And I think part of it is biochemical, and those components are different in sea serum at different concentrations, and there would have to be a lot more engineering involved to put them at a level, or to have them come out at a level that's appropriate. And frankly, we don't even know what those levels are now, because we need the individual patient studies to determine those. So that's the one challenge. But probably the one aspect of the study that I don't think has gotten a lot of attention, and I mean, I can say it now because it's been a few years. In a study like developing a brand new entity, and a latex skin test antigen, even though there were some, as Sandra said, other countries have them, for the U.S. that would have been a brand new product. And anytime you're developing a brand new product, you need a gold standard test, a test that you can rely on that says, as a tiebreaker, so to speak, this individual is latex sensitive or they're not latex sensitive. That test in the U.S. right now is a provocation challenge test, which basically means that as part of our study, in order to characterize whether or not the patients were truly allergic to latex, part of the protocol had them undergoing a provocation challenge, which meant taking latex allergen on powder in a room where it was suspended in the air, and they would breathe it in and have an appropriate reaction that confirmed their degree of severity or sensitivity to latex allergy. As the trial began to get into its important stages, some of the investigators that we were working with refused to conduct that test on their patients who were the most highly sensitive. Their pushback was that it was unsafe to do that, and we understand that, and we don't disagree with that. But the only way that we could have generated data that would have been relevant to licensing that product is to have the product tested on the most sensitive subjects that we could find, because that's, for the FDA's opinion, that's what defines their safety profile. And so we just could not come to an agreement with some of the investigators on how to do this both ethically and safely. And that, combined with the compositional challenges that we had, basically meant that we just could not find we couldn't find a clear way around those roadblocks, and that ended up pretty much slowing the study down considerably. I hope that made sense.
Yeah, thank you for sharing that. The next question is actually one that's very interesting and has come up now a couple of times in different questions. Based on your expert opinion, do you believe that GMO foods increase allergic reactions in latex allergic patients? And secondly, someone recently read that GMOs are used to make tomatoes stay fresher longer, and they could, again, potentially cause a reaction. What are your thoughts?
Yeah, I mean, I don't fall on either side of the GMO argument. I'm not a strong proponent of it. I'm not an opponent of it. I think it's a reality that, a lot of things that we don't even know are genetically modified are being modified now. We all want our produce to last for 3 weeks, but if this is a consequence of it, then there are some alternatives. There are heirlooms. There are some organic species out there. Sometimes they're hard to find. And the difficulty is that a lot of your grocers don't really know what's genetically modified or what's not. They can't trace the genetics of the particular fruit or vegetable to anything that would tell them that it's genetically modified or which genetic modification it has. Because of the types of molecules that are involved in latex food cross-reactivity and my understanding of what happens when you stress plants, I think it's very possible that the allergen levels of these particular allergens in plants and in edible foods can increase with genetically modified organisms compared to others that have not undergone these changes and are maybe less resistant to stress. But I don't think there's a lot of data on that yet. So I would caution against drawing what might appear to be a pretty obvious conclusion or pretty clear conclusion based on a little bit of information. I think there's still a lot more study that needs to be done to show exactly how risky some of these changes might be and whether or not there are other changes that are occurring genetically in these foods that might be helpful or beneficial in different ways. And so it may be, a bit of a trade-off that, if you're latex sensitive, it may be, a downside, but there may be more. Again, I'm not saying this is true. I'm just using an example. There may be more antioxidants or anti-cancer type compounds in some genetic organisms. So it may be a real mixed bag there.
Yeah. Okay, next question. We are coming up on the 8 o'clock hour, so we'll take just a couple more questions and then close out. Is there any funding for studying latex allergy now that you're aware of? Any new statistics? Also, this particular individual says that they are recognizing more latex allergy in young adults without the traditional risk factors.
Yeah, I wish I could say that there's a lot of investment, research investment going on in latex. Sadly, I think once the hospital environments went to latex-free environments and some manufacturing did as well, I think a lot of latex allergies sort of fell off the radar for a lot of folks, and it's clearly still a problem, and I think it needs to be the focus of more funding. One thing I always emphasize strongly when I speak to doctors and meet them at meetings and we have conversations like this, I encourage them to write up their case reports when they have incidents of their patients that have cross reactions, and in particular those that are maybe not quite as mainstream as the foods that we've talked about already and are fairly well established. I think that's one way to keep this subject in people's minds and remind folks that there's still a problem out there. It's a different problem maybe than we had 10 or 15 years ago, but it's still one that needs research, it needs funding, it needs attention, and I think it needs a combination of the physicians, the scientists, the manufacturers, the regulators. I think it's important to have all voices heard on this, and
including patients. Including patients, Dr. Greer.
Including patients,
yes.
Absolutely.
So
Absolutely.
Another question, in regards to a spina bifida patient who is only latex precautions, who's only taking those precautions but never had a reaction. There is not a reason to limit foods, but be aware that there could potentially be a reaction, mild reaction in the future. Is that the correct thought process?
Right. Yes, it is. It is. And there are a lot of individuals who may fall into these categories based on their either a blood test or their history, but they may have very low or no symptoms. And there are some folks that have very strong protective immunity against these allergic reactions. And it may be that they expose themselves to other types of allergens that carry similar structures, and they develop more protective immunity from it. It's fascinating to me that there's a pretty wide spectrum of reactions to these types of foods in latex-sensitive individuals, some more than others. And I think there was also a component of our clinical trial that we had a lot of difficulty clarifying for the FDA, because as we were trying to pursue a product, normally your subject group, or the groups, the individuals who would actually have your products used on them, have to be very well defined to the FDA so they can assess risk and benefit. Mm-hmm. And we had a tough time doing that because we were talking about a lot of different populations, and with the cross-reactive food question, there was still, back then, there was still a lot of confusion as to which particular allergens were involved, and whether or not some patients might naturally show more resistance than others. And so, yes, to answer the question, I think it's wise to be wary of those relationships, but it's very possible that the level of reaction to those foods would be low enough to where they could still consume them safely.
Okay. One final question. So certainly with all of the data that's been shared over the last 2 evenings and latex allergy awareness, but also the cross-reactive risk, isn't this a good basic reason to ban the use of latex gloves globally? And if we could, potentially limit the risk of those life-threatening reactions if you do have a position regarding the ban of latex, this person is asking for that reaction.
I agree. I know Sandra talked about this last night. There are many very suitable alternatives now to latex gloves. But those of you that have used latex gloves probably recognize the one characteristic that probably keeps them on the market, and that is the amazing tactile properties that they have. Mm-hmm. In terms of their comfort and the ability of the folks wearing the gloves to either operate or manipulate their hands much more freely and easily. The non-latex gloves are good, but they're not quite to the same level. So depending on how important that is to you, and some people claim that's a safety issue for them, that if they go to a non-latex glove, that they're putting themselves at risk because they can't feel their instrument or their tools as appropriately as they need to. And again, as long as they're not sensitive, that's not an issue. I do know some folks that used to wear latex gloves on the outside of a different type of glove on the inside, but that sort of defeats the purpose of in my opinion, there's no reason to wear 2 gloves when you don't have to. But I'm kind of with Dr. Gottschalk on this. I think the time has come for for alternatives to take the place of latex gloves. And I'd be curious to know who is currently buying the latex gloves, whether it's an occupation or an industry, and look at the sales of latex gloves and where they go, or if it's just homeowners that prefer their comfort over other types of gloves. And if that's the case, it may be and they're not sensitive to the latex proteins. It may be a difficult change. The other thing, of course, that I think is a factor is latex gloves tend to be less expensive, and in some cases that.
Well, that's one comment that was just made from someone in healthcare in California, that they are still very commonly used because of the cost there.
Right.
And then also wanted to just one other comment that came over regarding the GMO for tomatoes. That product was called Flavor Saver for shelf storage, and it's no longer on the market, according to a vegetable pathologist who is on the line.
Okay.
Let's see. We just a few more. comments here, and I think that we are wrapped up. A couple of comments in regard to the Massachusetts bill that's just been heard for food service limitation of latex 2 weeks ago, and then also in New Hampshire, there's a bill coming up that we can support. The network will certainly be following up and in contact in regard to how you can lend your voice to those particular advocacy issues. Many notes of thanks here from participants tonight. Dr. Greer, I could go on and share those with you. I think that, again, we just cannot get over the fact of what an excellent presentation this was and how comprehensive it was on the cross-reactivity and the concerns around latex allergy. And I'd like to once again just thank you as our presenter this evening for all the work that you did in preparing and presenting, as well as our sponsors, Mylan Specialty for the educational grant, the College of Allergy, Asthma, and Immunology as a co-host, as well as the American Latex Allergy Association. The Allergy and Asthma Network, along with the American Latex Allergy Association, are here to support you and help you through your journey. Please do not hesitate to reach out to our organizations if there is any way that we can support and help you along the way. Again, this recording will be provided for each of you for playback along with the slides, and we'll also be sending out a survey for your response tomorrow. Thank you again for your time this evening, and we hope everyone has a wonderful day. Take care.
Thank you, everybody.